Yes, thanks for having me. So yes, I'm Rebecca, and I'm an electron microscopy scientist at NeCEN. So I will start off with an introduction to NeCEN, our services, equipment, and then access modes, and then talk about some examples of tomography work that has been going on at NeCEN. NeCEN is part of the Leiden University Faculty of Science, and I know perhaps many of you were at NeCEN last year for this workshop, but in 2016 we were located temporarily here as part of the Faculty of Science. These buildings are now gone, and this building is down to about 2.5 stories left. They're slowly taking it down. NeCEN moved in, or since 2017, we've been located here in this new building. And the future plans for the Faculty of Science are to finish taking down all of these and then expand completely this entire building. So it's yes, a very nice building. We have a nice large open atrium. We're down in the back on the bottom, of course, but it's a really nice place to work and to come and do Cryo-EM. Okay, so our services, both Dutch and international research institutes, as well as companies, can come for services at NeCEN. And the services include sample preparation and quality assessment. So we do some freezing and screening on the spot. So this is mostly part of the INEX screening. And data collection as a service, data processing. Some companies come and we'll do some processing for them, but this is typically a collaboration with research groups. And then training and education, and a little bit of research and development through collaborations. So to do all of this, this is our laboratory equipment. We have an ML2 laboratory space, so we can work with a little bit more pathogenic things than maybe typically we could do. We have a Vitrobot, and then for grid storage, we have both a dewar that we use the normal Falcon tubes in, and then we also have a dewar with a puck system. So this puck system was developed in Ariana Riegel's lab at the Institute of Biology Leiden, and actually made through the fan mechanics workshop at Leiden. So it's a nice, more cost-friendly way to handle grid storage. And then the typical grid clipping and loading stations. We have two Titan Cryos microscopes. Our Cryos 1, this is also an ML2 in an ML2 lab, so we can image these samples on this microscope. It has a K2, and so this is kind of more our tomography microscope. And then the Cryos 2 has the Falcon 3, and it also has a CS corrector. So these are where we would do our high resolution data collection, and also training if someone comes for Cryos that wants to be a Cryos operator. We also have a Talos L120C. This is where most of the screening is done, and some training also on this microscope. And then finally the Akelos, which is also ML2, for preparing lamellae for Cryo-ET on the Cryos. And then looking at the overall workflow, we don't do protein preparation at NeCEN, although we have a facility in Leiden. But we would do negative stain and some diagnostic cryo-EM, often for companies. These would be done on the Talos, and then initial cryo-EM data collection, and also high-res collection on the two Cryos instruments. As far as training, we kind of do all types of training. We have a cryo-EM school. We've done 2 of these so far. The first was 9 weeks, the second 4 weeks. In the first one, there were in-person lectures, but with the second iteration, we went to the online lectures. And we go through basically the entire single particle workflow with this. So freezing on the Vitrobot, clipping, loading, doing a microscope check, and then setting up data collection with EPU. We've also done one-on-one training where someone will come for 1 week, and we do basically the same process, except with this we don't have this big final course exam where you go through the entire workflow. And then also just training for a specific task. So if a user wants to learn to clip grids, or they want to learn to set up data collection, we can do this pretty much at any step. So this is more where our Cryo-ET training would be, since it's a little bit more individualized. As far as user access, we have open access and then also reviewed access. For open access, you would apply through NeCEN's website, and this is evaluated based on the technical feasibility. The review is private and internal to NeCEN, and it's pay per use. And then for reviewed access, there are a couple of different ways to apply for this, through Instruct-ERIC, of course. So this is for researchers from Instruct member countries. The price is reduced, and there's no limitation on data collection time. And then iNext, so we have recently finished iNext, but this is for Transnational Access. The data collection and sample shipping are free, but it's limited to 2 days. And this is where we have done a couple of the screening projects. And then, as was mentioned earlier, iNext Discovery will be starting in February. So we'll continue with this, and I think we will also keep going with the screening work there. And then finally, the NEMI, so the Netherlands Electron Microscopy Infrastructure. And this provides data collection to Dutch researchers at a subsidized rate. And this is based on scientific excellence. Also, the NEMI provides funds for upgrading. So for instance, we will upgrade to the K3. That'll be installed next month. And we're planning to apply for a new Cryos as well. Okay, so I'll move on to the tomography, and I'll overview what we're doing at NeCEN, and then show some examples using subatomic averaging and Cryofib milling. Right now, well, actually over the past 2 years, our data collection projects have basically been 50-50 single particle and cryo-electron tomography. Almost all of our single particle data sets are being collected with EPU. I think over the past year, maybe 2 data sets they were done with Cerium. So it's single particles mostly with EPU so far. But our tomography projects are kind of split between different data collection softwares. So in 2018, the majority of our users were asking for tomography 4, a handful for UCSF tomography, and then very few Cerium. But we've seen a pretty good expansion in the number of users that want to use Cerium. And I think this is for a number of reasons that I'm sure many of you are familiar with. So people like to map things and see where they are and have a record of this and navigate. And then the flexibility of Cerium, so you can implement basically whatever you want to do, you can tell it to do it. And particularly as far as flexibility goes, they want to do the dosimetric tilt schemes. So I think this is mostly where we're seeing this. And then also the speed, you can get things going pretty quickly depending on how you want to collect your data. So there's a lot of control there. Okay, so this is an example from Ariana Brigel's lab. Again, this is work done by Wen Yang. And she was looking at Chemotaxis and E. Coli. So chemotaxis allows bacteria to sense chemicals in the environment and then move based on chemical gradients. And in E. coli, there are extended membrane-bound arrays of chemoreceptors. And the ligand would bind and then transmit the signal, which is relayed to the flagellum and can lead to a bias in the direction that the flagellum rotates so that the bacteria can change directions. And with this project, they wanted to characterize the different conformations of the chemoreceptor signaling molecules. And they want to do this in a near-native environment. So they used E. coli cells that had been gently lysed. So this took them from the native about 500 nanometers down to about 200 nanometers, and looked at them locked in different states, so in the kinase on and the kinase off, so that they can compare the two conformational states. So this was data collected on Titan Cryos. This particular project used UCSF tomography, and then proceeded with subtomogram averaging. So they were able to achieve 24 and 20 angstrom resolution maps of the two conformations, and the like biological relevance here, looking at the kinase on versus kinase off, they can identify differences in where these are dimers of trimers, where they are trimers of dimers, where the dimer splays, and also see weaker densities here indicating more flexibility. And then taking it one step further, they used these maps and did molecular modeling, so they fit predetermined crystal structures, and then also did molecular dynamics flexible fitting. So you can see in the video here the linear interpolation between the two states, and we see basically bending here. These are where a number of glycine residues are located. So it indicates that these glycines are facilitating the splaying, so providing a biological function. Okay, so the two of the major challenges with Cryo-ET are that it takes a while, so the data collection is relatively slow, you don't get as much data, and the size limitation, because some of the samples people want to look at are very large. So as far as the speed of data collection goes, recently people have developed, and we were able to test also on our microscopes, fast tomography. So in this, with this data collection scheme, you are continuously running your camera. You have calibrated the stage so that you know you don't have to do the focusing and tracking, and you can really shorten your data collection time. And some of the initial work was done only on single tilt stages where they were thought to be more stable, but our microscopes have dual tilt stages. And so we were able to test this with them and found very reasonable results. So it suggests this would be feasible on our microscopes as well. Unfortunately, I don't have any data to show yet, but hopefully that will be coming very soon. And then for thinning the samples, and I think this will be talked about quite a bit during this workshop, we can do Cryofib milling. So we have an aquilose at NeCEN, and this is just a schematic of how this would be done with a cell and then forming or milling a lamella so that you can see the internal components. This is in well, it was supposed to be a movie, but this is an example of data collected from a lamella, and these are bureau cells infected with equine arteritis virus. This is from Monza at the LUMC, collected on the Cryos with Cryos-EM. So you can see really beautiful structures within the cell, which we would not be able to visualize without Cryofib milling. But electron tomography is slow, so is Cryofib milling. So also recent work, these both came out on the same day, two methods for automated Cryofib milling, where you can use tracking and similarly to automated Cryo-ET, track the positions and automate how the lamella are milled. So this is one solution for speeding things up is automation. Another that has been worked on in NeCEN is trying to increase the throughput by keeping the lamella intact because these are very fragile. So as you can see, the lamella may bend, and when this happens as you're milling, you can accidentally mill areas that you don't intend to. So you would end up getting a large hole, or perhaps the lamella would just break. And this is work from Georg Wolf. So as you can see, the cryogrid films are subject to cryocrinkling. And when they're cooled, you go from a nice flat layer to a very wrinkly layer. And when this happens, well, let me move on. Sorry. When this happens, you may be creating forces that would lead to the lamella breaking during the cooling. Okay, so the solution here was to mill micro expansion joints on each side of the lamella. And I think you can see them here from the fib angle, and then from above in the TEM, the joints here. So these are just basically long spaces that are milled on each side, with the idea being that the tensions would be transferred to that space and creating a buffer so that your lamella doesn't break. The effort isn't increased much. You just add these during the first rough milling stage, and it would take 2 to 3 minutes per lamella. It doesn't add instability to them. They are not, falling out and breaking because of the gap. And with the particularly crinkled grids, the success rate goes from 23 to 78%. So it can be very helpful if you're having trouble with this type of breakage. And then this is an example from a tomogram collected from these lamellae. Okay, so future directions for these types of things at NeCEN. As I mentioned, we'll have the K3 installed next month. And we are really working towards implementing fast tomography. Another aspect that we would like to implement is the live preprocessing of tomography data. This will also be talked about, I'm sure, in this workshop. And then continue with the development of Cryofib workflows. We want to get this to the state where we can offer this as a service, and rather than just having specific users. So we would like to move towards this. And then also microelectron diffraction with fib milled crystals. So as you can see, the crystal here and the lamellae formed from the crystals, we would really like to implement this at NeCEN as well. And I went to the Diamond workshop 2 weeks ago. This is looks like really exciting and promising work, so we would like to start doing this. So I need to say thank you to everyone on the NeCEN team, and also like to advertise an open position for NEM scientist. So if you know anyone or if you're interested, please talk to Ludo. And thanks to all our partners, especially the work that I showed here from the LUMC and the IBL in Leiden, and all of the funding resources as well. And thank you.
Thank you. So we can start the discussion, I guess. Questions? You alluded to various topics, building, tomography, so
yep.
There are a lot of things to discuss there.
How do you find the ice film contamination in the equipment?
So we generally okay, I think, but we have had a few issues. I don't know if you have anything to say. No. I think it's generally okay. We don't have a lot of complaints unless something has gone terribly wrong, and yeah.
So about the tomography, you mentioned briefly that you were comparing single tilt and dual tilt. Is that, and how does it compare in your hands right now?
So we aren't able to do the comparison ourselves because both of ours are dual tilt. Mm-hmm. But with the work on fast tomography, they wanted to compare from their own microscopes the single tilt to ours. There was a bit of a jump at the 0 degrees going back and forth, but that can be corrected with calibrations. So it does seem perfectly reasonable to do.
So basically a good stage calibration will catch up errors. It depends how precise it can be, but it's true. Because it's a real question on knowing that now the developments are pushing into going only with single tilt
right.
acquisitions to make it faster and more precise, but on the other side, if we want to go towards unique structures, we cannot average. And we don't want to average. So we should be able to do a good dual tilt also. And I don't know what comments about that actually. So it could be really that the instrument becomes limiting at some point if we stick only to single tilt microscopes.
Well, you can still order the dual axis as an option. Just like the single tilt was an option, the dual tilt is now the option, right? So.
Yeah, sure, but then one should do something about making the onto more stable. Maybe one can do more about it, because the development on that are frozen more or less, not?
Yeah, but I mean, going from this dual axis to single axis, I mean, the big misconception is that Fred Jensen consistently called it a high precision stage, well, it's just a different holder.
Yeah, it's a different holder.
That's the only thing.
Yeah.
I really can't call that a development.
It's a different holder.
Well, I mean, that's the only thing that happened. Because they had the holder already and it gave good results of Antarctica and glacials. So it's not really a development. Anyway.
Yeah.
So I'm talking about the Titan Cryos, because for the Titan Cryos, for example, if you have the double axe, Thermo Fisher doesn't want to continue to support this. So they don't want us to do a double axe tilt acquisition.
Yeah, it's a question we are raising right now.
So yes, but we have to push them to.
Essentially, what we don't support is the software.
Yes.
But as you may, yeah. So at this moment, we have to make a choice. There is a guy over there developing this stuff, and we did decide to go for single axis because most people are more comfortable with that, and most people do sublimation. But it doesn't mean that in the future, we will not switch to something about the dual axis and support the tomography. Just that at the moment, we feel that the trend is that most people want to go to single axis, and we want to provide new software that works for them. Would that be okay for you?
Yes, but the software we have doesn't work.
That's right.
Manually,
Or you can use serial EM like they do at Nissan.
yes.
I have a naive question on this quick tomography. That what is the camera what you use? How do you use the camera there? I'm not really familiar with this.
So it's continuously collecting during the entire tilt series.
So it's not a Falcon, I guess.
No, this was done on K2 and also K3.
Regarding the zero tilt, so the jump there, I was wondering when we are talking about calibrations, what kind of exactly?
What kind of calibrations?
Yes.
So I don't know the details of how the calibration is collected, but you run basically a test tilt series ahead of time, and I think it uses the tracking to calibrate the movements, and then applies that to when you're collecting, so it would make the appropriate shifts.
So do you apply that then in your software, or is it done by Thermo Fisher, or?
So this is all run in serial EM, and
Okay,
yeah, so it's applied there.
You mentioned the 3D tomography software. How do you choose which one you use? Is it from the user demand or?
Yes, so our user would request what they would like to do, and it's...
And how long it takes to set up the serial EM?
Right, so it's typically based on their experience, what they have liked using before and what they have used before, so whatever they're comfortable with.
But you set it up for them, or they do?
Both, so we can set it up, or if they're experienced users, they will do it themselves.
Including for tomography,
Yes.
In single particle, which software do you use for image reconstruction?
So most of our data sets, the users will reconstruct themselves, and I don't know much beyond when the data goes out, so, but it seems, I don't know, it seems like people are using Relion, Cryospark. I'm not sure exactly what they're doing.
Do you do any online processing for tomography or?
We don't have anything implemented yet, but yes, we are aiming towards this very soon.
For high resolution tomography, the thinner the better. So you are talking about 230 nanometer thickness. Can you go thinner than that? And is there a limit? Because you are blowing a gallium beam and this heats up some parts of the section. So is there a theoretical limit that we cannot go thinner than that?
I don't know. Maybe the audience would have a better answer than I can do. Yeah.
Yeah, but the thing, just to animate that discussion, if it's very thin, then you won't see the neighboring objects. So at some point, you don't want to thin it too much either. It's a question whether it's good to actually go as far as technically feasible.
I mean, if you picture it in a way that material science does it, right, they can do much thinner in that sense. I would agree. If you cannot accommodate a single ribosome anymore, right, like 30, 60, it becomes problematic. But you can regularly hit definitely well below 120, and then the really thin ones are maybe like 50. These are ones really nice to work with.
Yeah, but you lose the information of the neighborhood, no?
Yeah, exactly. Exactly. But that's different applications, right? Well, depending on what you want. If you want the morphology, of course, you want something slightly thicker, but you know, subchromogram averaging definitely 100 and below.
Because for SEM, that would be interesting. So it's really different applications. Yeah.
So for fast series, how would you manage the tomography sessions? So typically, I mean, for one week, we will have like 5, maybe at most 8 to 10 lamella. Then they set up a session. Then afternoon they need to come back again and set up another session. And then the evening they need to come back again and set up another session. So how do you manage that as a facility?
Yeah, we haven't implemented the fast tomography data collection as a service yet, but yes, this is how things are working at the moment. Is you set up the session and then come back later when that grid is finished. But yeah, something we need to think about.
So there's, as part of INEX, there's also a wish of the community to provide more access for screening and that. So how do you see that? Because some of the centers are fearing how to deal with that, and how do you plan to organize that practically? Because it's very time consuming and needs a lot of optimization and so on.
Yeah. So for the past INEXT, I think we had 3, 4, 5 maybe screening sessions or projects, which were that was reasonable for us to handle. I'm not sure how many we're looking at going forward, but yes, it can be time consuming. We just.
But also the environment, I mean, the people on a facility, they will need to work almost full time on one single project. So that's, I mean, how realistic is that to really screen a lot of samples? That's a general question. Has that?
You say screening sessions. Are you talking about this enhanced support?
Access,
yeah. I mean, we got 2 days, which is just useless. Somebody comes with a proteins, you spend 2 days, you freeze a bit, and the second day at 5 o'clock, well, that's it, we did it 2 days, it didn't work. Have fun, next one. Utterly useless exercise. And this really needs to be discussed at some point. It's just a waste of time. Nobody gains anything from it.
Exactly, so that's why I'm starting the discussion actually. Maybe we should do much shorter sessions. I mean, in some cases, within the first hour, you already know that it's going. So I think we should do something about it.
Well, I always try to sell it internally as in, should we offer something as a service that we wouldn't do that way ourselves? Because they said it, they said, okay, let's do this internally with the students in the PRs. We train you in the Vittelborg, you get 2 days. If you don't manage, too bad. Nobody will go for that. But you offer it as a service. It's ridiculous. Completely ridiculous. It just takes time away, you don't gain anything.
Yeah, but
that's why
there's a way we could have shorter sessions and do training at the same time. Because then at least people can train in their home lab. I mean, and get the training to do that in the home lab.
The idea was that people don't have EM at home and a plunger. That was it. That's what it's for.
Yeah, but it's probably not realistic.
Okay, and we shouldn't do it, right?
Okay. Are there some other aspects? Want to require? Okay. And maybe also a common problem that our facilities have is to run on a long term. So how do you see that, for example, for upgrades? You mentioned briefly NeMe is supporting upgrade of the PS3. So that's very good. But is that something that is sustained on a long term? Can you plan things, or is that practically a new case? Just,
If you mean scheduling users during this time?
no, I mean to upgrade. Because one of the issues is to have a long-term support to actually be able to buy new equipment. Depending on the facilities or the countries, it's very different. So maybe you can say a word on how that is actually working practically. To get like new Cryos or things like that, so new microscope in general.
Yeah, I would have to defer to management here.
Yeah, so we are part of the NeMe, right? So as part of the NeMe, we're a national center providing access to our resolution machines. So in the NeMe consortium, we are applying now for a new machine. So just providing interest, but the way it's funded in the Netherlands is that you need support from all the NeMe partners, and that's what we're gathering at the moment. But that's a good point you're raising, and that sustainability is also an issue, and keeping up with the latest development. I don't think there's a one solution for everyone, so it's a case by case. That everyone has to do his own strategic development.
I have a very naive question. I always see the lamina is like rectangle. Is there any particular reason? I'm not really doing tomography. Any reason has to be always rectangle? And it's why is a single particle because it's really thin, like 10 nanometer. We don't see much breakage, but why that one is always break? Is there any reason, particular reason?
I think this is a much larger area of ice than would be within a hole, so it would be more subject to breaking. And for the shape of it, it's milled with two areas one above and one below the lamella. So you could shape it, I guess, any way you wanted to, but if you just have the two, one on top and one on the bottom, it's going to create a rectangle. Just intrinsically, that's how it forms. So.
Okay. Other questions? Seems to advance. Many thanks.
Thank you.